Well, hello, everybody. Thank you for joining us today in our live webinar. Today, we will be having the company of Stacy Wendell. It's going to present some of the results about bridging anti-inflammatory nitro-fatty acid signalings to metabolism. She's an associate professor at the University of Pittsburgh, and I'll be also your host in this session. My name is Andrea Lopez, and I am the head of the lipidomics facility at the Babraham Institute in the UK. This webinar is being recorded and it will be uploaded onto the Lipid Maps YouTube channel and into our Lipid Maps website within a few days. This webinar will take around 45 minutes with 10 minutes for questions. So please use the Q&A function for questions related to the presentation. And we will ask you to unmute yourself, or we will unmute you to be able to ask the question. We will take as many questions as time allows, bearing in mind that Stacy is in the United States, so it's close to lunchtime over there. Please also join us for the next free live webinar on the 25th of January, 2022. Will be presented by Associate Professor Katrin Washtinger, and registration is now open, and you can register in our Lipid Maps website. I would like now to introduce our speaker. I'm very happy to have met Stacy today. She's an associate professor in the Department of Pharmacology and Chemical Biology and the scientific director of the University of Pittsburgh Health Science, Metabolomics and Lipidomics Core. She obtained a PhD in chemistry from the University of Maryland, Baltimore County, and was an NRSA postdoctoral fellow in the laboratory of Ian Blair at the University of Pennsylvania, where she developed small molecule LC-MS methodologies for the analysis of environmental biomarkers of lung cancer and arachidonic acid metabolites. Stacy's current research is focused on the formation and signaling for bioactive fatty acids in asthma and inflammation. More specifically, Stacy's studies focus on how electrophilic fatty acids, such as nitroalkenes, promote cellular protective responses through the modulation of immune cell metabolism to alter inflammatory signaling pathways. Stacy, very welcome to our webinar, and please, the floor is yours.
Thank you, Andrea. Can everyone hear me okay?
Yes.
Okay, great. All right, so thank you for the introduction and the opportunity to present my lab's work. I'm going to start off telling you a little bit about what my lab research is, which the slide also serves as a nice outline for the seminar. So our basic overarching research area is electrophilic fatty acid formation and signaling. So these electrophilic fatty acids are those that contain either a nitro group on the carbon-carbon double bond or an alpha-beta unsaturated ketone. And we're interested in their ability to signal in inflammatory resolution as well as in metabolic homeostasis. And I'm going to tell you about two stories today. One, looking at how these electrophilic fatty acids and using nitro-oleic acid as our exemplary electrophile, how they modulate immune cell metabolism. And this is something that we've been working on recently. We've always looked at endpoints of inflammation and how they're altered with these electrophilic fatty acids. But more recently, we've been connecting immune cell metabolic status to these inflammatory outcomes. And the second part, this is a clinical translational study where we've been using nitro-oleic acid as a therapeutic in obesity-associated asthma. And I'll tell you a little bit about that story as well and how it took us in a completely different direction than we had originally hypothesized and led us down the road at looking at how nitro-oleic acid may modulate bile acid synthesis and metabolism in individuals with obese asthma. And so just to provide some background, I'm sure that many of you are familiar with this side of the slide, looking at how inflammation can give rise to oxidation of fatty acids with the formation of hydroxylated fatty acids that can be further oxidized enzymatically or non-enzymatically to alpha-beta unsaturated ketones, We've also recently looked at how these alpha-beta unsaturated ketones are further metabolized in a paper that just came out this year, demonstrating that in first pass metabolism, the enzymes responsible for reduction of double bonds do not actually inactivate the electrophilic moiety, but reduce other double bonds, leaving bioactive metabolites from these alpha-beta unsaturated ketones. So this is one area we're still exploring. But today I'll focus more on this side of the slide, which is demonstrating how we can give rise to endogenously formed nitrated fatty acids. And so these, as far as we know, are not formed through enzymatic processes, but through the modulation of inflammation and also dietary substituents. So alterations in diet, where there's an increased intake of nitrate from different vegetables and high nitrate containing foods, can lead to the reduction to nitrite by enterosalivary bacteria. So these are commensal bacteria that can reduce nitrate to nitrite. And in the stomach and gut, where there's a lower pH, can give rise to nitrous acid. We can also, through other pathways, form reactive oxygen and nitrogen species. And both of these processes can lead to homolytic scission and decomposition to form nitrogen dioxide, which is a radical that through addition reactions can form nitrated fatty acids. And so, as I mentioned, we're using nitro-oleic acid as our exemplary electrophilic fatty acid in our studies. However, we demonstrated not too long ago through murine studies and also in humans that nitro-conjugated linoleic acid is the primary electrophilic fatty acid that's nitrated endogenously. And we did this in a study where we used N15 labeled nitrate and nitrite, and we watched over time and monitored for the formation of N15 labeled nitro-conjugated linoleic acid. And we could see these levels rise very quickly after the administration of both nitrate or nitrite and conjugated linoleic acid orally. And we see that these levels rise to responses to levels or concentrations that are able to promote adaptive signaling responses in vivo. And so these electrophilic fatty acids, both the alpha-beta unsaturated ketones and the nitrated fatty acids, don't signal through G protein coupled receptors. They actually post-translationally modify reactive nucleophiles found. So either reactive nucleophiles found in transcriptional regulatory factors or redox enzymes. They can also form microaddition adducts with small thiols like glutathione. And so this nitro group here is very electron withdrawing. Just find my pointer. So this nitro group is very electron withdrawing, making this carbon electron deficient. And so it's very reactive with nucleophilic amino acids like cysteine. And in the case of the nitrated fatty acids, we form reversible microaddition adducts. As I mentioned already, these are naturally occurring. So not only do we find them endogenously in humans, but also in other species. Their levels can be modulated by diet and inflammatory status, and they link metabolic and inflammatory status to adaptive gene expression and protein function. And so myself and my colleagues have studied these electrophilic fatty acids in great detail in a variety of different disease pathologies. And two of our favorite endpoints to look at are the inhibition of NF-kappa B or pro-inflammatory signaling, and also the activation of the antioxidant response through NRF2. And so we know from other studies that have been conducted that we have alkylation of P65 of NF-kappa B, which inhibits DNA binding and inhibits the formation of pro-inflammatory cytokines and the expression of pro-inflammatory enzymes. Also, we can form a microaddition adduct with specific cysteines found in KEAP1, and KEAP1 traditionally holds NRF2 in the cytoplasm. Once it's alkylated by nitro-fatty acids, NRF2 can translocate to the nucleus to upregulate phase 2 responsive genes such as hemoxygenase, NADPH, quinone oxidoreductase, and others. And so just an example of this, we see here, these were alveolar macrophage taken at the time of a bronchoscopy and plated, and they were exposed to LPS. And so with LPS, we have an increase in cyclooxygenase 2 protein expression, which can be abrogated with the simultaneous addition of both nitro-oleic acid and the endogenously formed nitro-conjugated linoleic acid. We also see a concomitant expression of heme oxygenase 1, indicating that NRF2 signaling is present. We can also look by ELISA at cytokine levels. And again, we see this is in raw murine macrophage cells, but we can see that with LPS treatment, we have an increase in MCP1 and TNF alpha, which can also be reduced with the addition of nitroleic acid. And so, as I mentioned, we always look at these endpoints, but we never looked at how changes in central carbon metabolism might directly impact these inflammatory outputs that we always look at. And so we know very well now from the literature that central carbon metabolites themselves, depending on their cellular concentration and location, can also be pro-inflammatory. And so there's been a lot of interest in immune cells, especially at looking at levels of itaconate, succinate, and citrate, and how they may promote a pro-inflammatory or M1-like polarized state in macrophage and other immune cells like DCs and T cells. And so we thought to look at this ourselves and how nitroleic acid might affect macrophage polarization. And just to remind you of the TCA cycle or the Krebs cycle, during macrophage polarization to a pro-inflammatory-like state, there are typical breaks that we observe in the TCA cycle. And the first one of these breaks comes after citrate, where citrate can be shunted off out of the mitochondria and be used for de novo lipogenesis by ATP citrate lyase. There's also an upregulation in expression of cis-aconitate decarboxylase, or as also it's commonly known now as the immune response gene, which through this cycle, the break, instead of having carbons go through to alpha-ketoglutarate, we have formation of itaconate, which is then in part responsible for this break here, which inhibits succinate dehydrogenase and inhibits the oxidation of succinate to fumarate. And so we have a buildup of succinate. Succinate buildup can result in the stabilization of HIF1 alpha. We also, much like we see in cancer, during this pro-inflammatory polarization, have a reliance on glutamine, and glutamine gets shunted into TCA through alpha-ketoglutarate. And so we were very familiar with working with the raw murine macrophage, and so we thought to optimize our conditions to look at how our nitroleic acid would potentially alter central carbon metabolism. We decided to do this in the murine macrophage cells. And so we used 10 nanograms per ml LPS to treat the cells over a time course so we could determine which time was optimal for us to look at cytokine expression, as well as the max concentrations that we would reach with our pro-inflammatory metabolites of interest. We also chose to do this in both C12 glucose and also universally labeled carbon-13 glucose. So we could look at atomic percent enrichment and trace our labeled carbon through metabolism. And so all of this was run on a Thermo Fisher high-resolution IDX, which is a tribrid instrument, which we could look essentially a variety of different experiments. And what we chose to do in terms of this, and what we needed to do, what was sufficient for our data, was to look at targeted metabolite analysis. So looking at the absolute amounts of metabolites of interest, as well as carbon-13 isotope log tracer analysis. So looking at how many carbons were labeled in each of these metabolites as we go through TCA and other pathways. And so first, we wanted to look at, or confirm, I should say, that we could see reduction of nitric oxide formation by nitric oxide synthase. So it's been reported in multiple papers that nitroleic acid reduces INOS expression through its inhibition of NF-kappa B signaling. But this was really important for us to also demonstrate here, because nitric oxide and the buildup of NO can inhibit oxidative phosphorylation. And when this occurs in macrophage, we have an inhibition of OXPHOS, which leads to an increase in reliance on glycolysis. And so we can see here with LPS, we strongly induce expression of nitric oxide synthase, which is blunted with nitroleic acid. We can also look at the other product of arginine metabolism by INOS, which is citrulline, and we see here that LPS increases this metabolite, which again is blunted with a simultaneous addition of nitroleic acid. We can also use oleic acid, which is our non-electrophilic control. Interestingly, what we see here is with LPS plus oleic acid potentially increases the amount of citrulline that's present. And we also see this in another proxy that we can use to measure nitric oxide, which is looking at the amount of nitrite in the cell supernatant or in the media, where we see again that LPS increases the concentration of nitrite in the media. Again, this is abrogated with nitroleic acid, but potentially increased with a combination of LPS plus oleic acid at the same amount of nitroleic acid, which was the 5 micromolar. And so we also wanted to look in this case at our pro-inflammatory metabolites over our time course. And a lot of this work that I'll be showing you was initiated by James O'Brien, who was a medical student who had a year of research time in between his 2nd and 3rd year of medical school. And he was very productive during this time in the lab. He was assisted by Steve and Maddie, who were on the other side where I showed you the experimental design. They're part of the core facility here. And then he has since left the lab, and this project's been taken over by Alison Manuel, who was recently a postdoc in looking at post-translational modifications of glutathione in asthma. So she has a nice background for the work that we do. And she joined the core as a research scientist this spring. And so what we see here is in our time course, after our addition of LPS, we have an increase in pro-inflammatory metabolites. So we see this increase in lactate that we would expect with a reliance on glycolysis. We also see increased levels of succinate, itaconate, and glutamine. And we also see that over time, with the addition of the nitroleic acid that we have here in the red line, decreases in the levels of these pro-inflammatory metabolites. What's really interesting too, and something that we have yet to further explore, is that nitroleic acid alone reduces the amount of glutamine intake into the cell, even compared to vehicle treatment. And so this is something of interest to us that we will be further exploring in the future. But so from this data, we decided to choose our 12-hour time point. This was optimal not only for the metabolites that we were interested in investigating, both absolute amounts and through carbon-13 tracer studies, but also in the, which I'm not showing you, the amounts of cytokines produced in this study. 12 hours was also optimal to demonstrate their expression as well. And so what we hope to show in this work is that the effects of the nitroleic acid go beyond just inhibition of nitric oxide formation. And so we can see this here. It will become more evident on this slide and the following slides, where we're looking at the atomic percent enrichment of the carbon-13 label in some of our metabolites. And so just to show you what we did, we had universally labeled glucose that was all 6 carbons or C13. As we go through glycolysis, we form lactate and pyruvate, which both have 3 carbons labeled. And pyruvate can enter into the TCA cycle once it gets into the mitochondria through a number of different pathways. Well, not so many, I guess, but several different pathways that are more traditional. One is pyruvate dehydrogenase, which accounts for, in most cases, the primary route of pyruvate entering into TCA through acetyl-CoA. So acetyl-CoA is formed. Now 2 carbons can be labeled in acetyl-CoA. However, we can also have acetyl-CoA coming from other sources, such as fatty acid oxidation, but in this case, these carbons would not be labeled. And so acetyl-CoA combines with oxaloacetate in the first turn of the TCA to form citrate. Pyruvate can also enter in through pyruvate carboxylase, where pyruvate with carbon dioxide can form oxaloacetate, and in this case, 3 carbons would be labeled. And so this demonstrates here that there are many different labeling patterns that can take place with even the first turn and second turn and so forth until we reach isotopic steady state for citrate. So we can have completely unlabeled citrate, 2 carbons labeled from PDH, 3 carbons labeled from PC, or a combination of both in which 5 carbons are labeled. And so if we look here at lactate as a product of glycolysis, which is linear pathways, relatively simple. We can look at the isotopologues here on the x-axis. So this just indicates the number of carbons that are enriched with C13. And the reason if I didn't mention this before, the reason we have our unlabeled parallel experiment going on in the process is so that we can accurately background subtract out the natural abundant C13. And so what's really of interest here for lactate is looking at the M+3 isotopologue, where we see with LPS that we have an increase in this M+3 isotopologue of lactate. If we look at nitroleic acid here in purple, we see that this is decreased in comparison to LPS alone. And what was really interesting is when we started doing these studies, and as I mentioned, we wanted to look beyond inhibition of iNOS. We also had some experiments where instead of adding nitroleic acid, we added the inhibitor to iNOS 1400W. And so 1400W does not inhibit iNOS expression, but inhibits iNOS activity. So you have a decrease in the amount of nitric oxide formed. And what's really interesting is, although we're decreasing NO, which should therefore not inhibit electron transport chain and should not rely so much on, these cells should not rely so much on glycolysis, we actually see that there's no reduction in the M+3 isotopologue of lactate. And so we can start moving around TCA and look at other metabolites. As I mentioned, with this first break coming in macrophage, we see here upregulation of cis-aconitate decarboxylase, which leads to the formation of itaconate, which inhibits succinate dehydrogenase and leads to an increase in succinate concentration. So here we can measure itaconate. It's virtually non-existent when in cells that are not treated with a TLR4 ligand, such as LPS, or another toll-like receptor ligand. So here we see a very sharp increase in itaconate. This is again at the 12-hour mark. Levels of these are just relative amounts to internal standard, but it's been reported in the literature that itaconate levels reach millimolar concentrations, so much on the level of lactate concentrations in the cell. With nitroleic acid, we see a decrease in this pro-inflammatory metabolite. And what's interesting is we do not see this decrease with inhibition of iNOS by 1400W. Actually, again, much like the oleic acid, we see a significant, at least statistically significant, increase in itaconate with 1400W. This is recapitulated in our Western blot for cis-aconitate decarboxylase, or IRG1, where we see expression with LPS, diminished expression with nitroleic acid, but again, perhaps even an increase in expression with 1400W. And then if we look at succinate levels, so accumulation of succinate, which tracks with itaconate to some level, we see again an increase with LPS, diminished levels of succinate with nitroleic acid. And what's really interesting though is we see diminished levels of succinate with 1400W. So while it's thought that itaconate is directly linked to succinate accumulation, there must perhaps be other pathways involved in succinate accumulation that we're not taking into account for yet, since these increased levels of itaconate with 1400W are in opposition to the decrease in succinate that we see here. And so another way we can look at what's going on through TCA is looking at the fractional synthesis of succinate to glutamate. And so we can look at this by adding up the atomic percent enrichment of succinate, so all of the isotopologues for succinate M+1 to M+4, and adding up that enrichment and dividing that by the enrichment we see in glutamate. So if that enrichment is equal, we would have a ratio of 1, indicating that all the succinate that's formed is coming from glutamate, which is coming through TCA, based on our input of our labeled glucose. We see with LPS that this ratio goes down, which is indicative of a dilution in the succinate enrichment. And so this is very likely coming from exogenous glutamine that's either entering through GABA shunt or through alpha-ketoglutarate into the TCA cycle. And what goes along with what I described to you on the previous slide, where we see that nitroleic acid is actually able to decrease levels of glutamine alone, but also in conjunction with treatment of LPS, we see that this ratio is restored with nitroleic acid. So there's less of a reliance on glutamine in the presence of nitroleic acid. And so we also see that this decrease in succinate with nitroleic acid also affects stabilization of HIF-1 alpha. And so if we look at the protein expression, these are under normoxic conditions, but perhaps nutrient starved at the 12-hour point. We see that LPS increases HIF-1 alpha expression. And this is abrogated with nitroleic acid, but not so much with 1400W and certainly not with oleic acid. Again, this is just showing the succinate levels that we see here with nitroleic acid, again, reducing succinate. We can also look at IL-1 beta, and this is mRNA expression of IL-1 beta. And so we see with nitroleic acid, we have a reduction in mRNA expression of IL-1 beta, which would be a pro-inflammatory cytokine that we would see as a result of HIF-1 alpha stabilization. And so something we're working on now too is a Western blot for this to show the difference between the active and non-active forms of IL-1 beta with LPS treatment and with nitroleic acid. And so for this part of the work in the lab, some of the near-term goals are to discern the contributions to succinate accumulation by itaconate, as I demonstrated. 1400W does not necessarily inhibit itaconate formation despite lowering nitric oxide formation. So with nitroleic acid, this inhibition of succinate could be a result of the decreased unlabeled glutamine coming through GABA shunt or just coming in through alpha ketoglutarate. And so we have a colleague here at Pitt who has cis-aconitate decarboxylate knockout cells. And so we're going to be doing some work with them, looking at how these cells that can't make itaconate are affected by nitroleic acid to further tease out these pathways. We're also in the process of doing some glutamine tracing to further support some of what we see with the carbon-13 labeling from glucose. Another thing we'd like to do is examine the effects of nitroleic acid on M2 polarization. So can we either polarize macrophage to M2 with nitroleic acid alone, or can we induce macrophage plasticity? So it's been demonstrated in some papers that once macrophage polarized to a pro-inflammatory phenotype, this cannot be reversed, but perhaps with a therapeutic treatment like nitroleic acid, this could indeed occur since we see such a reduced need for glutamine in these pro-inflammatory cells with nitroleic acid. And so for the rest of the time, I want to move on to tell you about our clinical study looking at how nitroleic acid restores dysregulated bile acid metabolism, and these changes in bile acid levels are correlated to lung function, both in our murine model and in our clinical samples that we've been collecting in our obesity-associated asthma study. And so just to give you some background on obesity-associated asthma, asthma in general is a complex phenotype. So you can see here from this graph, we have severity on the y-axis and lifespan on the x-axis. And this is showing you basically two ovals that have been separated out based on cytokine profiles, so Th2 cytokines, which are, we think of as more traditional atopic asthma. And so these can be, for the most part, addressed with glucocorticoids. However, there's also a large cohort of non-Th2 asthmatics where Th1 and Th17 cytokines may predominate. Within this category and over on the adult side is obesity-associated asthma, although I would say at this point, in many cases, this oval could be flipped horizontal because with the epidemic of childhood obesity and other factors, we do see that there is many different ways that obesity can contribute to the asthma phenotype, and it's not very clear in some cases which may come first. And so I really like this Venn diagram that was published in this Jackie paper in 2018, which shows that there's overlapping phenotypes for obesity-associated asthma related to either asthma arising as a consequence of obesity, asthma phenotypes that are complicated by obesity, or uncontrolled asthma worsening obesity symptoms. And so we're not trying to focus right now on one specific boutique phenotype, but rather address the complexity of obesity related to lung function and airway hyperresponsiveness as a whole. And so we thought that the nitroleic acid would be a good potential therapeutic, as there are not many therapies for obesity-associated asthma, especially the non-Th2 obesity-associated asthma. And this is because colleagues have demonstrated previously that nitro-fatty acids modulate metabolic syndrome in a murine model of obesity-induced diabetes. So we see, again, reduction of pro-inflammatory cytokines with nitroleic acid from mice that are on high-fat diet. We also see increases in adiponectin and a concomitant decrease in leptin, and also in glucose tolerance tests, a reduction in blood glucose levels over time that are more in line with mice on normal chow compared to the high-fat diet controls. It was also observed in a phase 1 safety study where healthy adult males with higher BMIs, I believe the average BMI was around 29, so overweight to obese individuals who are otherwise healthy, they were given multiple different doses of nitroleic acid. So there was a 25 mg per day group and 150 mg per day group compared to placebo. So they were given this amount daily for 14 days, and different measurements were made. So here we see changes from baseline for leptin, MCP1, and IL-6. And this was somewhat expected. These were the inflammatory markers that we were interested in measuring. However, something that was unexpected was as these individuals were monitored over time out to 28 days, even though administration of the nitroleic acid ended on day 14, there was a substantial decrease in the amount of cholesterol, so total cholesterol, in those given the 150 mg per day dose of nitroleic acid, as well as a substantial decrease compared to placebo in triglyceride levels. And so with these changes in metabolism and all of the metabolic influences that and aspects of obesity-associated asthma, we thought perhaps targeting in a pleiotropic manner the inflammation as well as the changes in metabolic homeostasis with our electrophilic fatty acid might be a potential way to look at new therapeutics for this group. And so in collaboration with my co-investigators, Bruce Freeman and Fernando Holguin, we have what we hope, we're just waiting for the notice of award for our R61, so a clinical study where we're administering nitroleic acid, which clinically is termed CXA10 in individuals. So Fernando is a pulmonologist who's conducting the study out at University of Colorado, Denver, and sending us the samples. And so we will be, it's a double-blinded crossover study where we will have a nitroleic acid group and a placebo group, and then they'll have a washout period and switch groups. And during this part, we'll have some bronchoscopies where we'll be able to collect epithelial cells as well as bronchoalveolar lavage, but we'll also be collecting urine and stool and blood samples for all of our different measurements. In the meantime, what we've been working on while we've been waiting for this to take off is our murine model of allergic airway disease. And this is something that I've been working on in conjunction with a colleague, Michelle Manny, who's an expert in murine lung physiology and these types of models. And so what we've developed is a model of diet-induced obesity with allergic airway disease using house dust mite, which is environmentally relevant compared to the old OVA models that were often used. And so in this study, we have mice that are on just 60% high-fat diet or normal chow for 12 weeks. We sensitize them on 2 days, day 0 and day 7, with our house dust mite, and we use cholera toxin as an adjuvant. And then days 14 through 18, the mice are challenged with 2 micrograms of house dust mite. And it's during this time that we administer our nitroleic acid or our vehicle. And this is by oral gavage. And we do this prior to the challenge. So after the sensitization, these mice already have allergic airway disease. And so we're just administering our therapy during the challenge, and then sacrificing 2 days later after we take measurements for respiratory and mechanics. And so then these lung function measurements are done using what's called a flexi-vent apparatus, where the mice is basically ventilated and hooked up to this apparatus, which is measuring pressure flow and volume signals. And so we can measure resistance, which is an assessment of lung constriction, this elasticity or stiffness of that comes from the lung, chest wall, and airway, and compliance, which in some ways directly measures inflammation in the lung. So the lower the compliance, generally the higher the inflammation we see. And so what's really interesting, and I don't have time to talk about all of this, is that if you look at mice that are on normal chow versus high-fat diet, there are some distinct differences in inflammation. And in normal chow mice, in this allergic airway disease model, we see high levels of inflammation, so cell infiltration into the lungs that you can see, observe in the BAL, and look at the differentials and see the different types of immune cells that are present. We also see high amounts of protein indicating leakage. And we also see higher levels of airway resistance. And when we first started doing these studies in the mice on high-fat diet, we saw very blunted amounts of inflammation and not very amazing responses in terms of these lung function measurements. And at first we started to panic, and then we started looking at the literature, both for our murine model and for clinical studies that have been done up at University of Vermont by Matt Pointer and Ann Dixon. And we were then relieved to see what was really interesting is that when we look at individuals with obese asthma, they do have clinically blunted inflammatory status. And it seems to be that these metabolic changes that are going on are, although it's unknown how, are really contributing more to these changes in airway hyperresponsiveness than traditional inflammatory markers. So we were relieved to see that. And we do have changes in pulmonary resistance. If we look and see here, as these mice are on this flexi-vent apparatus, they're dosed with increasing concentrations of methacholine. So we do this methacholine challenge, which is similar to what you would do in the clinic to demonstrate presence of asthma. And so we increase our methacholine, and at 25 mg per mil, we see that our vehicle treated, so these are mice with allergic airway disease, have an increased amount of pulmonary resistance. So the higher the number, the more resistance compared to our control group. And our control group are mice that were sensitized with our adjuvant, but not with house dust mite. However, they receive house dust mite during the challenge phase. And so these are high-fat diet or obese controls. And then we also have our group that has allergic airway disease that's treated with nitro-oleic acid. And we can see a decrease at both 25 and 50 mg per mil methacholine of pulmonary resistance with our nitro-fatty acid. We also see increased airway closure in our vehicle treated group. So again, at 25 and 50 mg per mil of methacholine, with again, a significant decrease in the amount of airway closure with nitro-oleic acid. We can also look at pulmonary and tissue elastance. And again, we see at 25 and 50 mg per mil methacholine, significant decreases in pulmonary and tissue elastance with nitro-fatty acid compared to our vehicle control group. And so, as I mentioned, these results in our murine model correlate with clinical findings. And so this was taken from a paper that was published in 2014, where they looked at late onset asthma and saw little airway inflammation, but increased sensitivity to airway closure and elastance of the peripheral airways. And what was really interesting in this study is that they looked at individuals who were undergoing bariatric surgery, and they looked pre and post bariatric surgery. So there was a 12-month gap in between. So control individuals and individuals undergoing bariatric surgery who had asthma. And they were able to measure lung function in these individuals. And this graph is just showing E total, which is combined effects of peripheral and central airway elastance. And we see that bariatric surgery for both groups decreases this central airway and peripheral elastance, but also this change or the delta is more substantial in the group with asthma. And so for sure, there is some contribution of changes in metabolism, metabolic syndrome that have an effect on lung elastance. And of course, some of these could be mechanical as well, as increased weight on the chest cavity surely has an effect, but there's also effects of lung elastance in the distal lung, making those with asthma have lungs that are more collapsible. And so since inflammation wasn't seeming to be a main driver of what we were looking at, we were also interested in, well, how could these changes in metabolism be affecting the lung? So things that are happening at distal organ sites or within the gut microbiome. And so there's been many papers demonstrating that changes in gut microbiome or dysbiosis and the decreases in short chain fatty acids plays a predominant role in asthma and the development of asthma throughout childhood. And so we thought perhaps there were other metabolites that could be contributing to this phenotype as well. And in this paper and several others that we're looking at untargeted metabolomics and using this to look for changes in metabolism between different asthma endotypes, have always pulled out bile acids as potential metabolites that are dysregulated. And this paper that came out in JI several years back from Serpil's group at Cleveland Clinic was really focused on controls in severe asthmatics and showed from the work that they did with Metabolon that taurine, lathosterol, and tarchlorate, as well as some of the other bile acids, were dysregulated in those with severe asthma. And the one thing that they didn't really discuss extensively in the paper, but that was really of interest to me, was that when you looked at the BMIs of the groups that were used in the study, the controls had a much lower and statistically significant lower BMI than those in the severe asthma group, which were obese. And so we went back to our clinical samples that we had been saving in some of our studies and also in our mirroring model, and we started looking at all of the bile acids. And so what we're showing here is plasma bile acid levels that correlate with a decline in lung function as measured by FEV1. So the forced expiratory volume in 1 second. And so this is natural log transformed data. We had measured all the bile acids using LC high resolution mass spectrometry with stable isotope internal standards. And what we saw was that the glycine conjugated bile acids, so glycine conjugated cholic acid and ursodeoxycholic acid, were correlated with a decline in lung function. And this is data, these data points are all asthmatic individuals, so whether their BMI was below 25 or higher than 25. And we just looked at this as a measure of FEV 1%. When we pulled out those who only had a BMI greater than or equal to 25, the data stays the same for glycocholic acid, but the significance of this correlation increases for glyco-ursodeoxycholic acid, perhaps indicating that this bile acid is more associated with increased BMI. And so we looked at this also in our mirroring model of allergic airway disease. And in mice, there are the predominant bile acids that are formed in rodents are the murocholic acids. And so from our panel of bile acids that we were looking at, we saw significant increases in beta-murocholic acid and taro-beta-murocholic acid in both serum and lung tissue. And we could also correlate these with lung dysfunction. And so in this case, this is the correlation to some of those flex event parameters I showed you. So airway closure, which is G, or tissue damping. So we saw that increases in this parameter meant an increase in airway closure. So we see correlation of beta-murocholic and taro-beta-murocholic with airway closure, as well as beta-murocholic correlation with pulmonary elastance and pulmonary resistance. And so thinking about bile acids and obesity, we thought, of course, about looking at how bile acids are synthesized in the liver and potentially looking at gene expression to see if there's alterations taking place. And so, as many of you know, there are two primary pathways for bile acid synthesis, the classical pathway and the alternative pathway. And the enzymes, the cytochrome P450 enzymes that have an asterisk next to them also can be regulated by the microbiome. And so these were of interest to us to start teasing out, well, how does something that's changing at a distal organ site primarily have an effect on lung function. And so we looked at the gene expression in our mirroring model of some of these key players in bile acid synthesis. So CYP7A1, which is the rate limiting step in the first, in the classical pathway. And we see that we have our control mice here that are just our high fat diet controls. Our mice with allergic airway disease that are treated with vehicle have an increased level of CYP7A1 gene expression, which is reduced with nitroleic acid. Now, if we go back and we look at also CYP8B1, so CYP8B1 is responsible for the formation of cholic acid. And so we also wanted to look at this. We see here that this CYP8B1 levels in our mirroring model decrease, gene expression decreases, which leads to this ratio of, leads to an increase in the formation of chenodeoxycholic acid, which is actually a substrate for CYP2C70, and leads to the formation of the mirocholic acids in rodents. And so this would fit with our data showing that there were increased beta mirocholic acid and taro conjugate in the serum and lung tissue of the mice. And so again, this level is restored to levels of that's observed in control when in the mice that were gavaged with nitroleic acid. We also can look at a gene expression of enzymes that are involved in conjugation, and we see decreased levels of the conjugation enzyme with nitroleic acid compared to vehicle treated. So again, we have a decrease, potentially indicating that there's just less, that there's also a role probably for the microbiome here, and potentially something that we're investigating also with nitroleic acid and its role in modulating the microbiome and bile salt hydrolases that would deconjugate these, primarily in the rodents, these taurine conjugate bile acids. We also looked at one of the cognate bile acid receptors and the gene expression of this in the liver, FXR. And we see, interestingly, that although there's no change in the mice with allergic airway disease compared to control, that there is an increase in gene expression for FXR with nitroleic acid. So perhaps there's a feedback loop with FXR and CYP7A1 expression. So perhaps this is one way that nitroleic acid may be controlling bile acid synthesis gene expression in the liver. And so we think that allergic airway disease modulates enzymes to promote bile acid synthesis. So what's really interesting, right, is I told you that we can challenge, or we can sensitize these mice with the adjuvant and still challenge them with house dust mite, and they don't have allergic airway disease. And so it's only the mice that are sensitized with house dust mite and develop allergic airway disease that actually have these alterations in gene expression of bile acid synthesis enzymes. And so, of course, as I mentioned, some of these enzymes are controlled by the microbiome. So there's definitely a role for the microbiome in this, and just overall in changes in lung function related to obesity-associated asthma. And so we're also working on this angle as well, looking at how allergic airway disease changes gut microbiota. And so this is in collaboration with Barb Methe and Kelvin Lee. Barb Methe is one of the co-directors of the Center for Medicine and the Microbiome here at the University of Pittsburgh. Not surprising, there's a distinct difference between the gut microbiomes of normal chow mice and mice on high-fat diet. But what was really interesting to see in our preliminary studies is that these circles are showing you the distance between the gut microbiomes of pre and post allergic airway disease. And so we see here in the mice that are on normal chow, there's not really a difference at all in the gut microbiomes after we induce allergic airway disease with our house dust mite sensitization and challenge. However, we do see separation of the gut microbiome pre and post, also indicating that perhaps these changes in gut microbiome with allergic airway disease are altering bile acid synthesis. And so also preliminary data at this point, we see that treatment with nitroleic acid reduces the relative abundance of Acinospiraceae. And this is looking at those mice with allergic airway disease, either treated with vehicle or nitroleic acid. this is something that needs to be followed up on and further and investigated. And although what was interesting is in some of our modeling that we're doing with this preliminary data, we do see that actually Bacillus brachiae as well as Lactobacillus brachiae taxa are associated with increased small airway resistance. So with the Lactobacillus brachiae, there's negative correlation, and with the Bacillus brachiae, there's a positive correlation with airway closure. And so this working hypothesis is becoming a little bit too large for me, because I feel like there's a lot of different aspects involved. So we started off with the lung and always potentially thinking we were going to have to look towards the gut microbiome. But now we see potential changes with gut dysbiosis and with our model of allergic airway disease that are altering metabolism and synthesis of bile acids in the liver, potential leaky gut, which is allowing some of these bile acids into the bloodstream and their ability to travel to the lung. We are still working on what potential cognate receptors are expressed in the lung. There's not a lot of literature on this, as you might expect. Most of the receptor expression has been focused on the gut for bile acids, but there are several different potential aspects for transcription factors here to look at besides FXR and GPBR1, which is the G protein couple receptor. There's also the vitamin D receptor and NUR77, which is an orphan nuclear receptor. And so I just want to thank everyone for their attention, also acknowledge members of the lab who have done this work that I didn't highlight in the presentation. So Victoria Heinrich and Crystal Valle are doing a lot of the obesity-associated asthma work, as well as collaborators here at Pitt and at UC Denver. And thank you for your time.
Thank you, Stacy. Very comprehensive presentation of all your research and all the data. I'm going to encourage the participants to put the questions in the Q&A box. And I can see that we have one question by Valerie. So Valerie, would you like to unmute yourself?
Yeah. Hi there, Stacy.
Hello.
Can you hear me? You
Yeah.
can hear me okay, yeah. That was great, thank you. And I'm sorry I was a little bit late coming in, but the part of your talk there where you were talking about obesity and lung disease and bile acids, it's really relevant to the whole area of multimorbidity, which obviously, if you've got one kind of chronic inflammatory disease, you can be more prone to getting another one and then another one. And in later life, that may all be part of the kind of cognitive decline issues that people experience associated with multimorbidity. So I wondered if you'd kind of considered taking your model out in aging mice and looking at cognitive impacts maybe of some of this.
Oh, yeah, that's a great idea. I mean, for sure in aging, so these mice are, well, I guess in the aging world, these mice aren't so old, but in terms of traditional models used for asthma, these mice are definitely middle-aged at this point. By the time they're starting to undergo this like the endpoints of this model. But yeah, I think that's a great idea. One thing that we have been looking at too, which we just literally this week are getting the preliminary data for, is looking at how perhaps those with NAFLD may have altered bile acid levels that may impact lungs. So I was able to obtain some clinical samples from a colleague who runs a huge NAFLD clinical study, and we see some changes with asthma, those with asthma and NAFLD compared to NAFLD alone. So we've been going that route, but I think, yeah, for sure in terms of aging, and of course, that's a very hot topic area now. Yeah.
Okay, thank you.
We have another question by Chris Lynn. Chris, would you like to unmute yourself? Oh, he doesn't have a microphone, so I'm going to read the question for him. Have you seen any evidence of nitration of the itaconate double bond?
Yes, that's yes, because there are some papers that have shown that itaconate is actually a weak electrophile and can potentially activate NRF2. We have not looked at nitration of that, but that some of the studies that have been done showing its electrophilicity, it's definitely not a strong electrophile, and doesn't form a glutathione additive readily, but maybe perhaps, yeah, nitration of that would improve its electrophilicity. We have not looked at that.
Thank you, Stacy. We have a question in the chat by Stephen Barnes. Stephen, would you like to unmute yourself? Shall we try to unmute you, Stephen? I'm going to try to read his question because I cannot unmute Stephen at this moment. Haven't found how to unmute you. Let me see. I'm going to read the question. Haven't found the ability to unmute Stephen.
Can you hear me now?
Oh, yes.
Okay, great. Thank you. Stacey, I really enjoyed this talk, every moment of it. It's, the topics are very much to my heart. And I used to work with Bruce when he first discovered the nitro-lipids a long time ago. But I'm also a bile acid person. I'm glad you had you mentioned BAT, which is an enzyme we cloned and have studied a lot. I do work with the people in pulmonology here. And so one of the things they tell me, these are the clinical fellows, is that there is a problem of reflux. And the way they measure reflux into the lung is to measure torricolate. It's actually a very good measure of reflux because the bile acids, of course, are in the upper gut, released into the duodenum. And if they go back into the stomach and you have reflux, then they will actually go directly into the lung. So I wonder whether some of the effects are actually on a more direct route than rather than some of the more sophisticated ones that you've laid out. And
sure,
I was also curious, so this releases the other question, the scale of the changes in the bile acid levels. I did a lot of work many years ago developing bile acids as a way to examine hepatic function. So I worked in a liver disease unit in London. And so I'm just curious what the scale of, the bile acid level should be very low micromolar in normal people and normal mice. And so I couldn't, because I think you had log numbers, and I wanted you to translate what levels of bile acids are actually circulating in the plasma.
Yes. Oh, so yes, they are. They are very low micromolar amounts that are present in the bloodstream. Yes, that is true. And then going back to what you said about the reflux, I do think that's part of it also. Again, there are very few studies that look at GERD and the potential for bile acids there's some studies that have been done in epithelial cells treated with bile acids with that in mind. And they do promote inflammation. I think that's definitely a route that's of interest, and sometimes probably something we maybe forget about because, a lot of these people who are obese with asthma are on proton pump inhibitors, but that doesn't necessarily stymie the reflux portion, only the acid portion, right? So I think that's really something important for us to keep in mind.
Yeah. Okay. Well, thank you very much. Great talk.
Thank you.
Thank you for your question. We have another question in the chat from John Davies. Would you like to unmute yourself, John? John? I'm going to read his question. So he says, in the first part of your talk, you show effects on the TCA cycle. But do you know what happens to lipid metabolism? The TAGs, I imagine that means that triglycerols, or cholesterol increase?
So we have not looked at complex lipids. We do know, I actually took a slide out in the interest of time, where we do know that the nitro-fatty acid inhibits de novo lipogenesis through phosphorylation of ACC. Or we have preliminary data to show that. I shouldn't say we know definitively. And we know from previous studies that because of the inhibition of NF-kappa B and cyclooxygenase that we can inhibit formation of eicosanoids, which I guess depending on the situation could be good or bad. But we have not looked further and extensively into our lipidomics analysis yet to see if there are significant changes in triglycerides or other complex lipids.
He's also asking, does it also activate AMPK?
We do not know. We have not looked at that.
Thank you, John, for your question. Thank you, Stacy, also for participating in the last minute. I do have a question. So we were talking about this induction of glycolysis, which basically HIF is also the hypoxia factor, is also inducing this in terms of, for example, host invasion, viral invasion, or bacterial invasion. So do you think is that the nitroleic acids are playing a role into our immunity?
We have, yeah, I mean, we do think that we can modulate immune cell cytokine expression. So we have some preliminary work going on in dendritic cells where we know we can modulate expression of IL-12 P40 and other cytokines that would be relevant in terms of T cell activation. So we, yeah, we're getting there, but we're coming from a chemistry pharmacology to an immunology side, right? So we're getting there with collaborators and some things we're doing on our own in the lab, but we do want to go that route.
Fantastic.
Yes.
Very interesting. Thank you, Stacy. Thank you all the participants for attending today. And I will remind you, 25th of January 2022, we have our next webinar. Please be tuned to it. And lovely to see you, everybody. Thank you, Stacy, again.
Thank you, everyone.